Hello everyone, and welcome to today's live webinar, a workflow for CRISPR Cas9 high-throughput arrayed screening with synthetic CRISPR RNA. I am Maren Mayer-Gross of GE Healthcare, and I'll be the moderator for today's broadcast. This webinar is presented by LabRoots and sponsored by GE Healthcare. GE Healthcare continues to be an innovator in the field of gene editing and CRISPR Cas9 research, launching the very first synthetic CRISPR Cas9 solution, and now offering one of the most comprehensive gene editing portfolios in the industry. GE remains focused to develop effective tools for the greater research community. For more information about GE's CRISPR Cas9 solutions, please visit www.gelifesciences.com/CRISPR. Questions can be submitted anytime during the event by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will answer as many questions as we can. All questions not answered during today's event will receive an email response from one of our technical support scientists. The presentation window can be enlarged by clicking on the screen icon in the lower right-hand corner. If you experience any technical problems viewing or hearing this presentation, please click on the support button at the top right of the presentation window, or submit the problem through the green Q&A button. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive these credits. I would now like to introduce today's speaker, Louise Baskin, Senior Product Manager for GE Healthcare. Louise joined Dharmakon RNA Technologies in 2005. Since then, she has led the development and commercial launch of multiple genome-wide siRNA and microRNA product lines. Currently, she is responsible for developing and expanding the Dharmakon Editor Genome Engineering portfolio, which you will be hearing more about today. I will now turn it over to Louise for the presentation.
Great. Thank you, Maren. Good morning and good afternoon, everyone. Thank you for joining us. I'm going to be speaking today about high-throughput arrayed screening with synthetic CRISPR RNA libraries. So just as a quick review of the topics I'm going to cover, I will start with a brief overview of loss-of-function screening, and then talk through the recommendations and methods for carrying out a successful CRISPR Cas9 screen using arrayed synthetic CRISPR RNA. I'll then go through an example of how this can be applied for the purpose of RNAi hit validation. And lastly, I'll show you some results from various high-content assays with a sneak peek into a screen that we're currently working on. And then I'll close with a review of the tools that are available for you to do an arrayed CRISPR screen of your own. Now, most of the time when you think about loss-of-function screens where we knock down or decrease a gene's expression to uncover its function, our minds tend to go to RNAi screens. The use of arrayed siRNA or pooled shRNA reagents to carry this out has become really very mainstream over the past 10 years. In short, an siRNA is delivered into the cell where endogenous RNA interference machinery uses that siRNA to identify and degrade a particular messenger RNA, thereby decreasing its expression. But now that CRISPR Cas9 tools are becoming more and more prevalent in these loss-of-function workflows, it makes sense to utilize them also in these larger screening situations. Now, as a reminder, CRISPR Cas9 works through a very different pathway and requires not just an RNA, a guide RNA, to specifically target a particular gene, but also requires the introduction or expression of a Cas9 nuclease. And then these two components work together to target and cleave the gene at the DNA level. Once the Cas9 nuclease creates a double-strand break, the cell's own DNA repair mechanism kicks in. Non-homologous end joining, or NHEJ, typically results in slightly imperfect repair, where the site of cleavage will have mutations due to insertions and deletions, or indels for short. So when you're doing a gene knockout experiment, the desired end result is an indel that results in a mutation that is sufficient to render the resulting protein useless, like a frame shift or some premature termination. Now, while there are other repair pathways possible, for the purposes of today's topic, we'll only be covering results of non-homologous end joining to result then in a knockout of the target protein. Now, even though the CRISPR Cas9 tools are only a few years old, they have already been numerous screening publications, including whole genome screens. But to date, all of these studies have utilized lentiviral single guide RNA pools as opposed to today's topic of arrayed CRISPR RNA libraries. Now, I'm not going to do a deep dive into the differences between pooled and arrayed screens. Much like choosing between RNAi and CRISPR, the choice between pooled and arrayed screening may depend on tools you already have at your disposal, the experimental system you're using, and the biological question you're asking. But just in short, pooled screens, which are carried out with lentiviral reagents expressing an sgRNA or a single guide RNA, generally require less infrastructure as they are performed in large culture dishes. And because you're using lentiviral transduction, these can also be carried out in difficult to transfect cells. Whereas arrayed screens may require high throughput equipment if the library has hundreds or thousands of genes and potentially multiple reagents per gene. And generally, these are used with cells that are amenable to transfection. But while there's a trade-off in the amount of handling required, there's also a trade-off in the types of assays that can be used. Pooled screens require a method for selection of a phenotype that will either enrich or deplete a population of cells under the experimental conditions. But in contrast, there's really no limitation on the types of assays that can be used in arrayed screens. So this is just a more visual demonstration of what I'm talking about. Although arrayed screens will support very straightforward phenotypes like viability and drug resistance, they also expand the types of experimental questions that can be addressed into more visual cell-based assays. So these sample images on the slide are taken from an article on high-throughput RNAi screening, and they really represent a lot of high-content assays like translocation of tagged proteins, analysis of nuclei size and shape, morphological changes, up and down regulation, or relocalization of a protein. And there's really more examples than what I can name here, but the point being is there are opportunities for multi-parametric and very complex biological questions to be addressed when you're working in a one gene per well format. So then in bringing this concept back to the available screening tools, the most practical tool for investigation of hundreds or thousands of gene targets in an arrayed format is a synthetic CRISPR and tracer RNA, so this kind of dual RNA system. Now you may know that Dharmacon products are based on high throughput, high quality RNA synthesis, so it was a very natural evolution for us to develop our gene editing platform with synthetic RNA as the basis. So the editor, synthetic CRISPR RNA, is modeled after the naturally occurring dual RNA system, which includes the target-specific CRISPR RNA, which will couple then with the tracer RNA, which is the longer RNA that together forms a complex to activate the Cas9 nuclease that directs the cleavage of the genomic DNA target. So just a little bit more about the editor synthetic platform. Dharmacon was the first to employ the synthetic dual RNA approach for CRISPR Cas9 guide RNAs when we launched these editor products in 2014. And not only does it mimic the natural bacterial system, it's also much more practical for chemical synthesis. And now you might say, well, why does it matter if we're using synthetic when there's so many sources for expression plasmid or in vitro generation of guide RNAs that are available? Well, first, the editor RNAs arrive ready to use, having undergone full quality control. There are no cloning steps, no requirement for sequence verification, no purification or quantitation that needs to be done before use. These hands-on steps might be fine if you're working only with a few genes, but for dozens or hundreds of plates, you're at very high risk of introducing variation, and some of that perceived cost savings will really start to dwindle when you take into account the consumables and the hands-on time that would be required. There are also no design steps required with editor. The products are available pre-designed with an algorithm that selects for functional and specific target sites. Because the system is RNA-based, it's fully transient. There's no DNA components that might integrate into your cells, giving you less chance of off-target effects from any ongoing expression. And these small RNAs are also much less toxic than in vitro transcribed RNAs, and they're much easier to transfect than plasmid DNA. And since the CRISPR RNA is amenable to high throughput synthesis, as I mentioned before, we can very easily support applications like arrayed screening that require hundreds or thousands of unique CRISPR RNAs. Now, without doing a deep dive into the editor algorithm, I do just want to show you a quick couple pieces of data that illustrate the rationale behind it and why algorithm-designed CRISPR RNA is important. So using a functional knockout system, generating data from over 1,100 CRISPR RNAs across 10 genes, one of those genes is shown here, we identified a set of rules to select the most functional designs, not just the potential creation of indels. Now, I'll be discussing the details of the assay in a moment, but for now, just know that in this graph, the vertical axis represents functionality, so low to high, and the x-axis is every possible CRISPR RNA for every PAM in this gene's coding region. So the variability that you see of the functionality along the coding region really illustrates the importance of not just selecting a guide RNA in an early exon. I've listed a few examples of the selection criteria we examined and were found to be important. And then all of these functionality rules have since been validated in multiple systems and have been incorporated into our algorithm. Now here's one of our validation experiments where we turned to next-gen sequencing to detect insertions, deletions, and mutations at the CRISPR cut site for CRISPR RNAs with either the top 10 functionality scores for 10 different genes or for the lowest 10 functionality scores across those same 10 genes. Now, although this isn't functional phenotypic data, we do know that protein knockouts that arise from CRISPR Cas9 gene editing must be due to an indel. Therefore, we expected that our algorithm would demonstrate differences in the percent indel formation for CRISPR RNAs with either high or low functional scores. And what we found is that for high-scoring CRISPRs, 93% had greater than 40% indel formation, as you see in the upper graph, while only 33% of the low-scoring CRISPR RNAs were able to reach that 40% editing efficiency. So really demonstrating that CRISPRs with high functionality scores have higher overall indel formation efficiency. Okay, so I've introduced the synthetic CRISPR RNA system and the editor algorithm for functional designs. So now let's talk about actually how you'd go about doing an arrayed screen with these tools. So the four primary workflow considerations I will address today are here on the screen. First is selection of the source of the Cas9 nuclease. Second is optimization of delivery or expression of the Cas9. Third is optimization of the CRISPR tracer delivery. And then lastly, optimization of assay conditions. So you'll probably have noticed that the word optimize appears quite often in this list of considerations. And this really should come as no surprise. So for anyone who's done siRNA screens, you'll know that nearly every cell in the well can be transfected and will achieve 70 to 90% target knockdown. So this is really good penetrance, and it makes it relatively straightforward to call something a hit. With CRISPR Cas9, given that there are two required components, the Cas9 nuclease and the guide RNA, depending on conditions, you may get anywhere from 25 to 75% of cells being edited, and of those, not all edits will lead to a functional knockout. So we certainly hear concerns that this potentially lower penetrance could delay adopting arrayed CRISPR screening, because in an arrayed format, since in a high throughput setting, there is no opportunity to do enrichment of the edited cells. That's why it's really important to optimize delivery of both the CRISPR components, expression of the Cas9, and making sure that you're working with guide RNAs that give you efficient functional knockout. The quantitative reporter assay I referred to earlier when showing you the work we did with algorithm development is a functional readout involving disruption of proteasome function. We've used this assay extensively with siRNA, so we had a very good idea of signal strength and good positive control genes to work with. So we used an engineered U2OS cell line that has a mutant ubiquitin fused to EGFP, such that when the proteasome pathway is functional, the ubiquitin gets degraded and there is no GFP signal. However, when the proteasome function is disrupted, either by knockout or knockdown of proteasome components, we get an accumulation of GFP and then we get a signal. We've used this assay for a few different gene knockout studies in working with the CRISPR components. But before carrying out a much larger screen, we knew we had a bit more optimization to do. So let's start by going through my list, beginning with how we selected a source of the Cas9 nuclease. So there are many reagents available for gene editing using CRISPR Cas9. Again, what's required is the Cas9 nuclease and a guide RNA. So for the guide RNA, there's a variety of reagents that can be used, but as I've already discussed, we're going to be talking about today the synthetic CRISPR and tracer RNA approach. So now it comes down to the Cas9. Cas9 nuclease can be delivered as lentiviral particles expressed from a DNA plasmid or directly delivered as a protein or mRNA. But in the context of screening, rather than just doing a few experiments, you have to consider which source is going to give you the highest consistency across wells, especially over a period of time, while keeping in mind that co-transfection will introduce a source of variation. You also need to ensure high expression of the Cas9, since this will generally correlate with more efficient target gene cleavage. And then lastly, like everything you do with screening, you need to consider the cost of scale-up. So although Cas9 protein as well as mRNA demonstrate really good results when delivered with synthetic guide RNAs, it would probably be cost prohibitive to use protein when transfecting hundreds or thousands of wells. So with those caveats in mind, we started by working with stably expressed Cas9 cells using our editor Lentiviral Cas9 nuclease, demonstrated on the left, as well as testing co-transfection of the CRISPR tracer RNA with the editor Cas9 expression plasmid, illustrated on the right. Now, after doing the same transfection with both systems on 5 different days, we looked at fold change of our GFP readout in our proteasome reporter. Now, as you can see on the left, we had much higher fold change and much better day-to-day consistency when using the stable Cas9 cell line and then simply transfecting the synthetic CRISPR tracer RNA. Whereas co-transfection of the Cas9 plasmid with the guide RNA, shown on the right, was much less consistent and really didn't give as effective results overall in terms of high GFP expression with this positive control. So we went ahead with the Cas9 expressing cells. So now that we had decided on using stably expressed Cas9, let's look at the next step of then optimizing the Cas9 nuclease expression. So the basic workflow we used is illustrated here. Transduction of the lentiviral Cas9 nuclease. Those cells are then treated with blastocidin to select those that are resistant and therefore expressing Cas9, and then growing up and expanding that cell population. Now, we chose to continue with a mixed population rather than doing clonal isolation, but either method can be used. We have heard from other screeners that they've isolated clones, grew them up, and then assessed them for optimal cutting efficiency. The potential risk could be that there's an integration effect from where the lentiviral Cas9 integrates into the genome, but that would be diluted out when using a mixed population. In either case, it's just critically important to make sure you get good Cas9 expression. And that's where the editor lentiviral smart choice promoter selection comes in. So with the editor lentiviral Cas9, you're able to select one of 6 different promoters to drive the Cas9 expression. Now here are some results showing a promoter optimization study. Now in all cases, these cells are expressing Cas9 under the control of each of the indicated 6 different promoters, and then the cells were selected with plasticidin for at least 10 days before doing the transfection of the CRISPR tracer RNA. So here we used one of our assays, positive control genes, VCP, transfected the CRISPR tracer RNA in triplicate into the cells, and then after 72 hours used T7E1 mismatch detection assay to quantify the actual gene editing or indel formation. Now as you can see in the calculated relative frequency of gene editing, we got optimal cutting when Cas9 was being driven by the CAG promoter. The efficiency there was much higher than with other promoters. So from this experiment, hopefully you can see that this can be a very important step in maximizing gene editing, and we'd certainly recommend this to anyone who's developing a Cas9 cell line. Now one quick point I do want to make about mismatch detection assay results. It is generally understood that the percent indel formation is underrepresented by as much as 50%, just due to limitations of the sensitivity of the enzymes. But even with this limitations, it is effective to use in cases where you need to understand the relative efficiency differences as opposed to strictly quantitative. So now that we've identified the best promoter to drive Cas9 expression in our stable cell line, we need to move on to the second component of the CRISPR experiment, which is the delivery of the synthetic CRISPR tracer RNA. So using our Cas9 expressing proteasome reporter cell line, we transfected CRISPR tracer RNAs targeting 2 different positive control genes in increasing amounts, and then quantified the editing efficiency by looking at the percentage of cells expressing GFP. Now as you can see, we saw a plateau around 10 nanomolar concentration, and we didn't really see improvement as things went higher. So our general guideline is to work with 25 nanomolar concentration, because you really don't want to be on the low end of the working range, but rather more in the middle. And as with siRNA screening, one of the most important aspects of optimization is the transfection step. You need to make sure you're maximizing delivery of the RNA while maintaining cell viability and ideally keeping the amount of transfection reagent to a minimum. So for all of our experiments, we worked with the DARMAffect transfection reagent formulation that had shown good siRNA delivery results in these cells. Generally, the conditions for transfection of siRNA are going to be closely matched with the best conditions for CRISPR tracer RNA delivery. So although the screen results I'll be showing you later were done with forward transfection, we know from our extensive interactions with screeners and our own experience screening that reverse transfection is often preferred. So I thought it might be of interest to show an example of that here. So what you're seeing is our results of optimization of reverse transfection, first using a CRISPR tracer RNA targeting PPIB, where that knockout should really have no impact on viability as it's a housekeeping gene. So we used 3 different cell densities. We'd recommend testing at least 2 in your system, along with 4 to 5 different amounts of transfection reagent while keeping the CRISPR tracer RNA amount constant. So the upper image is looking at the viability of our cells in each of these conditions. As is often the case, as you increase amounts of transfection reagents, you can have a detrimental effect on cell viability. So the green outlined conditions are those that we found were best for each cell density. And then the lower image is the assessment of editing efficiency, again using a DNA mismatch detection assay, for a subset of those corresponding conditions above. It's really not going to do us any good to have happy, viable cells if we're not also getting good editing with those conditions. But as you can see, the green outlined results, again, were very good for both viability and the indel formation. Okay, so once you have a set of candidate transfection conditions, the final step is to assess all of this in the context of your phenotypic assay. Now, we start working with a positive control for our proteasome assay when we're doing this step of optimization. So here we're using PSMD7, which is a known positive in this gene, or excuse me, in this system, as opposed to a housekeeping gene like we use for the transfection optimization. So it is important to test whether those optimal transfection conditions are also optimal in the context of your assay. So in this slide, I'm actually getting back to the forward transfection conditions that we used in our screen. Here you're seeing 4 different amounts of the Darmeffect 4 transfection reagent, measuring both viability, shown by the blue dots, and fold increase of GFP expression, shown by the green bars. So by looking for the conditions that maximize both, we identified the optimal assay conditions, again outlined in green, and these are the conditions we used moving forward with the larger screen. So here's a closer look at the phenotypic and editing efficiency data we were able to achieve with our positive controls following assay optimization. On the left, you see treatment with either a CRISPR tracer RNA or an siRNA pool, which we'd run alongside some of these initial trials just as comparison. Targeting either PPIB as a negative control for our assay or PSMD7 and VCP as positive controls. Now, as you can see from the GFP expression, the penetrance is really pretty comparable and certainly robust enough for positive hit calling of this phenotype. And then on the right, you're seeing the corresponding gel, again looking at mismatch detection, indicating really very efficient gene editing for these assay conditions. So here's our final workflow. So the first step is, again, to generate the Cas9 expressing version of our proteasome reporter cell line. And we did this with the editor Lentiviral Cas9 nuclease. And as I mentioned before, we continued with a mixed cell population, but you may choose to develop and characterize a clonal cell line. So to set up the screen, we plated our Cas9 expressing cells into our assay plates, where they were allowed to adhere, sort of rest and recover for 24 hours. Then in a separate plate containing our CRISPR RNAs, so the experimental designs as well as positive and negative controls, we added the tracer RNA and then added the Darmeffect transfection reagent. Now I want to point out here that there is no requirement for a heating and cooling step to anneal the editor CRISPR and tracer RNA. They will hybridize at room temperature in just a few minutes. So then once those RNAs have had a chance to complex with the transfection reagent, about 20 minutes, this mixture is then added to the plated cells and the incubation time begins. For our assay, we used a 72-hour time point, which happens to be the same as what we'd used for an siRNA screen using the same assay. So now just to show you the slight difference in using reverse transfection instead of forward transfection, you would start by dispensing the CRISPR RNAs from the original storage plates into your assay plates, and then add the tracer RNA and then the Darmeffect transfection reagent to each well. And then after that 20-minute complexing time, the Cas9 expressing cells are added to those assay plates, and then the incubation time begins. So now, while not all cells are necessarily amenable to reverse transfection, it does save you time in the workflow and requires less handling of the RNAs. And here's the normalized triplicate results for one of our 8 plates. We screened 61 genes with 10 CRISPR RNAs per gene in triplicate. Now, 10 is more than what we would recommend now. At the time, we'd not yet finalized our guide RNA algorithm. We wanted to make sure that we were getting enough functional data points. But as you can see, when compared to the positive controls in green and the negative controls in red, we saw very nice clustering of phenotypes for both moderate and very strong hits, as well as some genes that were clearly negative in this assay. So following this screen, we felt really confident that working under optimized conditions, the phenotypic penetrance for the purpose of hit calling was really sufficient and wasn't going to require any enrichment of the edited cells. In this screen, of the 61 proteasome-related genes we used, we found 40 hits, which was pretty well aligned with what we expected from our prior siRNA screening results. And that will actually take us into our next topic. So what I'd like to show you now is a hit validation study we carried out to determine whether arrayed CRISPR RNAs could be useful as follow-up to siRNA screen results. So this is a data set of an siRNA screen we carried out a few years ago in this same proteasome reporter cell line, this time targeting over 5,000 genes. So shown here is the EGFP fold increase values from the primary screen, where the purple data points are positive controls and candidate hits, and the blue are negative controls and genes that were negative in the assay. So our primary screen was run with both siGenome and OnTarget Plus SmartPool siRNA reagents, and then we carried out additional confirmation with all 8 individual siRNAs that are components of the pools. So to look at the use of CRISPR RNA as a follow-up tool, we selected a subset of our hits as well as some non-hits to analyze with CRISPR knockout. Now, as you might be able to tell from the gene list, those non-hits were selected because they were known proteasome-related genes, but they were negative in the siRNA screen. So what we wondered was whether these genes had some kind of a threshold effect, such that unless they had 100% knockout as opposed to a 70 to 80% knockdown, might they still remain negative in the assay? So like with the CRISPR RNA screen I just described, we were still in a pre-algorithm phase when doing this work. So we were working again with 10 CRISPR RNAs to target each of these genes. As you'll see in later slides, we now recommend 4. So in this screen, we ended up working with 330 CRISPR RNAs targeting these 33 genes. Now, I'm not going to dig into all of the results, but I do want to show you a few representative genes that illustrate what we saw. So here's an example with one of our known positive control genes, VCP, where on the left, you'll see that for all 10 RNAi reagents that we used, the siGenome SmartPool, the 4 siGenome individuals, the OnTarget Plus SmartPool, and the OnTarget Plus individuals, they all gave a GFP readout above the hit cutoff point, so as expected, a very strong, high confidence hit. This was confirmed then by our CRISPR RNA results, where all 10 gave a similarly strong phenotype. Now here's an example of confirmation of a non-hit. So this gene was negative in our primary siRNA screen, but it is a known proteasome component, so we put it into our test as a potential false negative. However, as you can see, 0 out of our 10 CRISPR RNAs gave us a phenotype, so we feel pretty confident that there wasn't an issue with the loss of function threshold effect for this gene, but instead it just might not be expressed in this cell line or is not required in this cell line for the proteasome function that we're assessing. And then the last example I'll show you was really kind of a weak RNAi hit that would potentially sit on the bubble of whether or not we would proceed with it. So again, on the left, you'll see that only 3 of the 10 siRNA reagents have a phenotype, and not even a very strong phenotype at that. But when we moved to the CRISPR validation, we had 9 out of 10 designs give us a phenotype. So here might be a case of where the CRISPR RNA might have rescued this hit, kept it on the list a little bit longer as a gene of interest to do further studies. Okay, since I'm not going through every gene, I just wanted to provide a summary of what the results looked like. So everything in green on the table is a gene that had concordant phenotypes between the siRNA and the CRISPR RNA. So 10 of our 16 hits were confirmed, and 15 of the 17 non-hits were verified as non-hits in this assay. Now, there are probably a few different ways this can be interpreted, but we really feel that in general this is a very logical extension of the RNAi screening workflow. So to illustrate that, here's a workflow diagram of the screen that I just described. So an siRNA screen with around 5,200 genes is carried out with 2 SmartPool siRNA reagents per gene. Following confirmation with positive hits, we narrowed down to 26 candidates. Again, that's where we rescreen with any reagent that gave us a hit in the primary. And we then deconvoluted each of those pools to their 4 component individual siRNAs, giving us 8 additional data points. Now, this use of multiple siRNA reagents per gene is really considered best practice to verify an RNAi phenotype. So this then took us to 16 potential hits, where we used a cutoff of 3 or more positive siRNA reagents. Now, at this point, sometimes screeners aren't always sure what to do next. They might find a small molecule that targets their gene, but those aren't always available. So using CRISPR-mediated gene knockout is really a great method for orthogonal hit validation, since you can get away from the RNAi pathway and any of its potential off-targets and really just look at true loss of your target gene's function. So the next thing I'd like to show you are some results we've gotten with a number of different assays using now the 4 algorithm-designed CRISPR RNAs that are part of our editor library products, which I'll be showing you in a minute. So here are some results from an APOL1 assay, which, if you're not familiar, is an enzymatic assay measuring caspase 3 and 9 as an indicator of apoptosis. Now, even though cell death as a phenotype doesn't necessarily require an arrayed format, one of the most common questions we get is whether viability-related genes or essential genes like PLK1 will give visible cell death in an arrayed CRISPR workflow. For many people, this is a very popular transfection control, for instance. So what you see here in the graph are the caspase readout levels showing the expected increase for these 3 genes, which are known to induce apoptosis. And the images along the top of the slide are the well images under the microscope of the PLK1 results alongside a non-targeting control. So as you can see, for all 4 PLK1 wells, so each one representing a different algorithm-designed CRISPR RNA, the cells are kind of balling up and floating away and generally very unhappy and most definitely undergoing apoptosis. This next assay is with a similar set of genes as we just saw, but instead we were looking at a marker for mitotic index. Now these cells were again treated with the 4 pre-designed editor CRISPR RNAs. During meiosis and mitosis, histone H3 is specifically phosphorylated, so the assay we're looking at used an antibody for phosphohistone H3 and then visualized it with the in-cell analyzer. So the graph indicates the increase we saw for that phosphorylated histone H3 with our negative controls as well as our positive control genes, PLK1 and KIF11. And the images along the top are the cell images for the KIF11 knockout wells. So the blue stain is the nuclei and the red is the anti-phosphohistone H3 antibody. So again, 4 different designs for KIF11. All showing a very nice increase in the red stained cells that indicates the increase in mitotic index. Now the last set of images I want to show you requires just a little bit of background on the assay. So this assay is actually part of an ongoing screen we're doing with genes involved in the cell cycle. So we're using a G1 to S cell cycle phase marker reporter cell line. These cells have an EGFP reporter fused to a localization domain of helicase B. And helicase B localization is dependent on its phosphorylation state, which is cell cycle phase dependent. So as you can see in the diagram, during G1, it's localized in the nucleus. In early S phase, it gets phosphorylated and translocates to the cytoplasm. So this is a really good example of an assay that absolutely requires a 1 well per gene format that can interrogate genes, in this case, that may play a role in this aspect of cell cycle progression. So we're still doing our assay validation, but here are some really nice results with, again, our positive control genes. So note that rather than showing you 4 CRISPRs per gene, I'm showing you 1 CRISPR for each of 4 different positive control genes. So two things here on the slide. The EGFP localization as seen under the microscope, and then the cell cycle analysis based on that localization is shown in the pie charts. So sort of working left to right, when you compare to the non-targeting control, where we find the majority of cells are in S and G2 phase, you'll see that for cyclin E knockout, we get a very high increase in G1 cells due to that gene's known role in blocking G1 to S transition. And then for the next three genes, cyclin B, PLK1, and KIF11, which block mitosis, you'll see both from the pie chart as well as in the cell images that there's an increase in mitosis indicated by the nuclear EGFP. So as I indicated before, this assay is the basis for a much larger screen we're doing with our editor cell cycle CRISPR RNA library. And we'll be talking more about that as time goes on as the screen is completed. So what I've shown you so far today are a variety of phenotypic assays that have been successfully applied with arrayed synthetic CRISPR screening. The proteasome reporter assay, the APO1 enzymatic assay, and then two different high-content assays, one with that antibody for phosphorylated histone H3, and one with that EGFP translocation reporter cell line. Now, as you saw, under optimized conditions, all of these assays gave us sufficient penetrance to evaluate target gene knockout phenotype in a single well with no enrichment required. And so I really feel that these demonstrate nicely the potential for arrayed screening using the editor synthetic CRISPR and tracrRNA to RNA system. Now, lastly, before I take questions, I do just want to tell you a little more about the tools that are available for supporting all of the workflows that I've covered today. So first is the Cas9 nuclease. We offer 4 different formats of Cas9. The one most discussed today was the Lentiviral Cas9 using a constitutive promoter, but there's also an inducible version available. We've found this to be very popular with many CRISPR screeners because they worry about detrimental effects on their cells by having this constant presence of the Cas9. So with inducible, you get the power of stable expression, but it only needs to be induced and expressed just prior to the screen. For transient expression of Cas9, we offer 2 DNA-free options that can be co-transfected with the synthetic CRISPR tracer RNA. So we offer either mRNA or a Cas9 protein. Our Cas9 protein has a nuclear localization signal and is now available at a higher concentration than where you'll find elsewhere, which can be very advantageous when you're doing electroporation. And then lastly, we also have Cas9 as a DNA expression plasmid, which has the same choice of constitutive promoters as I discussed for the Lentiviral Cas9. And you also can choose between a fluorescent or an antibiotic resistance marker for when you do need to do a selection or enrichment of transfected cells. So now we get to the arrayed libraries themselves. The predefined catalog libraries that are available for human and mouse are listed here. You'll probably see some very familiar collections if you're all familiar with the sRNA libraries. And many of these human libraries are already made and on the shelf. Some of them, and including all of the mouse ones, would require a few weeks of production time. And we are currently working towards building a human druggable collection, which will be around 7,500 genes when completed. Now here's just a few more details on those catalog libraries. So they include 4 different CRISPR RNAs per gene, and they're formatted in 96-well plates with the outer columns left open for your assay-specific controls to be added. They're available as 0.1, 0.25, or 0.5 nanomoles per well. And of course, if you require more material, we would simply provide more replicates at those amounts. And since you will need to add tracer RNA to each well prior to transfection, we are offering bulk discounted pricing on the tracer RNA when you're ordering CRISPR RNA libraries. Now, if you're interested in carrying out siRNA hit validation, like I showed earlier, or maybe you have your own set of genes you want to investigate that don't overlap nicely with a catalog collection, we'd recommend a cherry pick library. So what you're seeing here is a screenshot of our online cherry pick library plater where you can configure your own custom plates. So with this offering, you're able to very easily upload your own gene list for human or mouse targets and then add the pre-designed CRISPR RNAs targeting those genes to a custom plate. We'd certainly recommend 3 to 4 CRISPRs per gene, but we do have up to 5 available in our predefined collection. The minimum order is only 20 wells, and you're able to add editor controls to the plate, as well as customize the plate layout however you like. The quantities available per well will range from 0.1 to 2 nanomoles per well, so there's really plenty of options for however many cell lines or however many assays you might be working with. To support these applications, you're going to find pretty extensive online resources to help you get started. Just a few examples are shown here. So one is the reverse transfection protocol that I referred to earlier. We also have a little more in-depth application note describing the optimization of reverse transfection. We also have a technical manual for doing creation of a stable Cas9 cell line, as well as carrying out that promoter optimization. And if you happen to be working with transient Cas9 rather than stable expression, we also have guidelines for optimizing co-transfection of the Cas9 mRNA or Cas9 protein with synthetic guide RNAs. And with that, I will wrap up with a quick summary before moving on to questions. Today I've shown you that it is very possible to achieve good loss-of-function phenotypes with no requirement to enrich for edited cells. The examples there shown with the editor synthetic CRISPR RNA. We do recommend stable Cas9 expression to improve reproducibility and penetrance, and to use the editor algorithm designed CRISPR RNAs to improve the chance of having multiple designs that give you functional knockout in this arrayed format. I reviewed data from arrayed CRISPR RNAs used to validate hits from an siRNA screen. We saw very good concordance across the data, and since siRNA and CRISPR tracrRNA have very similar transfection conditions and in our hands had very similar assay time points, there really shouldn't be a really high barrier to entry for this method if you're looking to adopt arrayed CRISPR RNAs. And then lastly, I showed you some snapshots of our positive control genes in multiple assays, including a few high-content assays, to really try to demonstrate the potential applications for arrayed screening with synthetic CRISPR RNAs that just wouldn't be possible with a pooled screening approach. And with that, I will thank you for your attention. I do want to remind you to visit our website where you'll find the products and resources I've talked about today. And now I'll turn it back over to Maren to start our Q&A.
Louise, thank you for that informative presentation as usual. Now, while we are getting ready for the Q&A session, 2 polling questions will appear on your screen. We really appreciate your answers to these questions so we can follow up appropriately with you. During the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Again, we will try to answer as many questions as we can. And it looks like our first question in the queue is, when you optimized with Cas9 plasmid, why didn't you wait 24 hours before transfecting the guide RNA instead of co-transfecting? Wouldn't it give you better results?
Oh, okay. So it sounds like you're referring to the method of letting the Cas9 plasmid kind of getting a head start on being transcribed and translated into the protein before adding the CRISPR and tracer RNA. So with co-transfection, it is certainly possible that there is some degradation of the CRISPR tracer going on while it waits for the plasmid to become a protein. That's not actually something we've seen a lot of in our own hands. But that's kind of separate from what you really need to think about in the context of doing an arrayed screen. it would not really be practical to do 2 rounds of transfection when you're handling a lot of different plates. You potentially have some toxicity problems. You'll definitely have inconsistency between wells and potentially inconsistent expression of the Cas9, and you're also doubling your cost of transfection reagents. So really between just kind of hands-on time and handling as well as overall efficiency, we would still recommend this stably expressed Cas9.
Thanks for that clarification. Another one of our viewers asks, what about cellular responses to stable expression of Cas9? Isn't it detrimental to the health of the cells to constantly have nuclease expressed?
That is it's a great question and a very, very common concern. There seems to be a very wide range kind of across the spectrum on this topic in terms of the level of concern we see or has been reported. I think the best answer is really that it's going to be cell type dependent. And you are probably going to be the best judge of whether Cas9 is causing any changes in viability or morphology or some other behavior. In our hands, the stable Cas9 version of the U2O cell line that I showed you today continues to grow and behave very normally. But we have heard from other folks who are making Cas9 cell lines that, for 3 out of the 4 they look fine, but for in the 4th, their cells just are starting to act strange or the morphology is changing. So certainly a concern. One way you can get around this, if this is something that you may observe, is that you could use the inducible version of our lentiviral Cas9. So the nuclease is only getting turned on just prior to the guide RNA being presented, and then it's time to cut the DNA and then it's turned off. Or you can use co-transfection of the transient Cas9. For cost purposes, I'd probably recommend mRNA over protein. They both give very good results. Just with a caveat in mind that co-transfection could induce a little more variability into the screen.
Oh, excellent. Thanks for that. Let's see. The next question in the queue is, what was the time point used in the mitotic index assay?
Oh, okay. I'm just going to flip through my notes really quick to make sure I get that right. That actually brings up another very common and very good question about time points. The logic might tell you that a Cas9-induced knockout should take longer because you're working at the DNA level compared to RNAi, where you're working at the messenger RNA level. You're kind of further down in the expression pathway. But we actually see that most of our assay time points between RNAi and CRISPR are very similar. I don't necessarily have an in-depth explanation for that, but that is what we observed. And to specifically answer your question, for the mitotic index assay, that was a 48-hour time point. So post-transfection of the CRISPR RNA was when that data was collected.
Very good, very good. Another question from one of our viewers is, how much do the CRISPR RNA libraries cost?
Okay, I didn't know we were going to go for cost questions, but all right.
Sorry.
No, it's okay. I'm not trying to dodge it, but I will have to say it varies, right? Depending on how many genes are in the library and whether you get a small quantity or a large quantity per well, obviously that's going to vary. I think that the easiest way to answer is that we've aligned the pricing of the libraries very closely with the price for an on-target plus set of 4 sRNA library. So 4 on-target plus sRNAs is very close to the price we're offering for editor CRISPR RNAs. The one thing you would need to keep in mind is that you would still need to buy the tracer RNA, but generally it's going to depend on the number of genes. But we have, like I said, aligned it very closely to our specificity-enhanced sRNA library prices.
Thank you. Let's see. The next question is, how many screens can I complete with one CRISPR RNA library?
Good question. It is also going to be dependent on how much you order, right? So how much per well. There is a calculator on our website in the Tools and Calculators section that can kind of help you determine this. But I'll give you sort of one method as a rule of thumb. For the smallest size that we offer, which is 0.1 nanomole per well, that is enough material to carry out 40 screens when you're working at our recommended 0.25 nanomolar in a 96-well reaction. So that obviously doesn't take into account, pipetting loss or anything, but kind of a rule of thumb, working at 0.25 nanomolar in a 100 microliter reaction, you can get 40 screens with 0.1 nanomole.
Oh, excellent. Thanks for that response. And now I'm being told that we are getting close to the top of the hour, so it looks like we have time for just one more question. Let me see. It is it also valid to use siRNAs to validate CRISPR Cas9 screen hits?
Oh, okay. Yeah, great question. So what I showed you was using CRISPR to validate siRNA. So then, obviously people starting with CRISPR screens might say, well, how do I validate? So obviously you'd still want to follow kind of the same best practice of using multiple reagents, per gene to make sure that there isn't some unique aspect of those CRISPRs that give you the phenotype. But I think it would be perfectly valid that the reverse would be true, that knockdown could serve as a validation method for knockout. I mean, it's potentially assay dependent, but I think it's a perfectly logical place to start.
Thank you. Thank you. And again, thank you to our audience for all the great questions, and Louise for your answers. If you have submitted a question through the Q&A button and it was not answered, someone from our technical support team will respond to you via email. If you have further questions that you did not submit during this broadcast, please email them to ts.dharmakhan@ge.com. And now, on behalf of GE Healthcare, we would like to thank LabRoots for making today's educational webcast possible. And this webcast will be available for on-demand viewing through December of 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.